Okay, let's unpack something truly fundamental. Where did we, as complex eukaryotic life, even come from? We tend to think of ourselves as the basic units, little self-contained worlds. But what if that picture is incomplete? Missing some key players we've maybe historically overlooked? I'm talking of viruses, those tiny genetic packages that usually get all the blame for making us sick. And then there's the big jump to eukaryotic complexity itself, the nucleus, mitochondria, all that intricate machinery. It feels like two separate puzzles, right?
Oh,
But our source material today suggests they're, well, deeply connected, almost two sides of the same ancient coin.
It's fascinating, this evolving perspective. We're really moving away from just seeing viruses as, pathogens. The more we learn about their ancient history, their biology, the clearer it becomes they've been active participants in the story of life for a very, very long time, playing roles we're only now beginning to fully grasp. Our source today really shines a light on this interwoven history between viruses and the cells that make up all complex life, including you.
Exactly. So our mission in this deep dive is to explore that connection. How could these tiny, often vilified entities have been so central? Perhaps even acting as, like, architects in the evolution of the complex cells that make up everything from a mushroom to, well, us. Get ready for some surprising insights that might just flip your understanding of life's origins on its head.
If we zoom out to the grand scale, we're really asking how biological complexity itself arose, and it's looking like viruses, often considered to be on the fringes, might have been right there in the thick of it.
So let's start by getting a better handle on our key player, the virus. We all learn the basic definition: obligate intracellular parasites. They need to hijack a host cell's resources to replicate. But our source points out that this definition is just, well, the tip of the iceberg. They are ancient, and they've been evolutionary powerhouses for eons.
That's a crucial point. They aren't just passive hitchhikers. Their dependence on host cells creates this constant evolutionary pressure, and their knack for moving genetic material around makes them incredibly influential. The discovery of giant viruses is particularly illuminating here. These viruses have genomes much, much larger than typical viruses, sometimes even rivaling bacteria. Incredibly, they carry genes for functions we usually associate with cellular life, including some bits of the machinery for making proteins. This really blurs the lines of our traditional definitions and forces us to reconsider what exactly is a virus. Think of it like finding surprisingly intricate clockwork inside something you thought was just a simple spring.
It really does make you wonder. Now, a fundamental question arises. When did viruses even appear on the scene? Were they before, alongside, or after the first cellular life?
Yeah.
Including LECA, that last universal common ancestor.
This raises a really important question with big implications. The fact that viruses can infect organisms in all three domains of life, archaea, bacteria, eukarya, that strongly hints at a very early origin. It's quite possible they were around even before these domains diverged from LECA or maybe emerged very soon after. There's even a fascinating idea that virus-like entities might have existed in a pre-cellular RNA world, that hypothetical time when RNA molecules, not DNA, were the primary carriers of genetic information and could even act like enzymes.
An RNA world with early viruses made of RNA. Wow, it's a wild thought. Now, our source outlines the main hypotheses about how these viruses might have actually originated. Let's start with the virus-first idea.
Right, the virus-first hypothesis, or primordial origin. It suggests that viruses, or at least virus-like self-replicating molecules, arose from complex organic matter before the first cells even existed. The idea is that these early replicators might have eventually figured out how to exploit the first cells, or maybe even played a role in their formation. Some theories even propose that cells arose as a kind of protective container for these primordial viral entities.
So in this scenario, viruses are almost the starting point. But our source also highlights a significant hurdle with this idea, right? How do you get the relatively complex structures and replication methods we see in viruses today without any pre-existing cellular machinery to build upon?
Exactly. That's the challenge. It's hard to imagine how intricate viral protein coats or the mechanisms for hijacking cellular processes could evolve in a vacuum without cells already being there. That's a key reason why the virus-first hypothesis, while intriguing, faces some skepticism. So yeah, a key takeaway here is that the complexity of modern viruses makes a purely pre-cellular origin a bit of a puzzle.
Okay, so if not virus first, what's the next leading idea? That's the escape or progressive hypothesis.
Yeah, the escape hypothesis. It proposes that viruses originated from bits of cellular genetic material, things like plasmids or transposons. These mobile genetic elements within cells that somehow manage to, well, break free. These escaped genetic elements would have then acquired the genes necessary to build a protective protein coat and other components needed to travel between cells, essentially becoming autonomous infectious particles. This idea also suggests a polyphilic origin, meaning different groups of viruses could have sprung up independently from different cellular hosts at different times. And the fact that some viral genes show similarities to these mobile genetic elements within cells offers some supporting evidence. Think of it like a piece of software code breaking free from a program and becoming its own independent app.
That makes sense. A rogue piece of cellular code going viral, quite literally. But the source also mentions a difficulty here. What about those ORFAN genes?
Or ARFN genes, yes. These are genes found in an organism that have no detectable relatives in other organisms, including cellular life. Many viruses have a surprisingly large number of these unique genes. And the escape hypothesis kind of struggles to explain where these completely novel genes came from if viruses are simply escaped bits of existing cellular DNA or RNA. So these genetic orphans pose a bit of a mystery for the escape theory.
Right, these genetic orphans don't seem to have any cellular ancestors. So that brings us to the reduction or regressive hypothesis.
The reduction hypothesis offers basically the opposite perspective, that viruses evolved from more complex, possibly even free-living cellular organisms that gradually lost genes and functions as they adopted a parasitic lifestyle. Over vast amounts of time, this streamlining through parasitism would have led to the highly dependent entities we recognize as modern viruses.
Okay, so almost like reverse evolution, going from complex and independent to simple and dependent. And our source mentions a more nuanced version of this idea, the ancient cellular origin model. That sounds intriguing.
This is a more recent model that's gaining traction. It suggests that modern viruses are the result of reductive evolution, not from a single cellular ancestor, but from multiple ancient cellular lineages. These hypothetical ancient cells, sometimes called virus cells, are thought to have had segmented RNA genomes and coexisted with the ancestors of modern cells. The idea is that these virus cells, due to the intense pressure of their parasitic lifestyle, gradually shed their cellular machinery, eventually leaving behind the diverse array of viruses we see today.
And the evidence for this comes from looking at the structures of proteins, is that right?
Precisely. This model is strongly supported by structural phylogenomics. That's a field that analyzes the evolutionary relationships of protein three-dimensional structures, known as fold superfamilies, or FSFs. Think of FSFs like fundamental architectural blueprints for proteins. Researchers have discovered a widespread sharing of these ancient FSFs between viruses and all three domains of cellular life, archaea, bacteria, and eukarya. This is known as the ABEV group, and it involves a remarkable 442 shared FSFs. This suggests a common ancient toolkit with proteins and the coexistence of these ancient virus cells alongside the ancestors of modern cells. So in this view, viruses aren't just recent evolutionary innovations, but rather preserve molecular remnants of an ancient form of cellular life that didn't directly lead to bacteria, archaea, or eukarya as we know them. They're almost like living fossils from a long-lost branch of life.
Wow, viruses as echoes of a lost world of cellular life, that's a powerful image. Now this brings up a fundamental debate. Are viruses monophyletic, meaning they all descended from a single common ancestor, or polyphyletic, arising multiple times independently?
This has been a long-standing question. The traditional view, favoring polyphily, largely stems from the incredible diversity we see in viruses. They have different types of genetic material, RNA or DNA, single or double-stranded, a huge variety of shapes, different ways of replicating, and, as we mentioned, lots of those enigmatic orphan genes that don't seem to connect them. The classification system used by the International Committee on Taxonomy of Viruses has historically reflected this idea of multiple independent origins for different viral groups. It looks like many separate evolutionary experiments all arriving at a similar parasitic solution.
So it seemed like a lot of separate evolutionary experiments that all ended up with a similar parasitic lifestyle. But the source says that analyzing protein structures is challenging this view.
Exactly. Protein structures tend to be conserved over much longer evolutionary timescales than the underlying DNA or RNA sequences, especially in rapidly evolving things like viruses. By looking at these highly conserved protein folds, researchers have identified a core set of viral protein folds shared across very diverse viral groups, even those infecting hosts from all three domains of life. This has led to the concept of an ancient viral supergroup, suggesting deep, unifying evolutionary connections among many viral lineages that we couldn't see just by looking at their genetic sequences. So, this structure-based approach is revealing ancient homologies, like finding the same fundamental tools in different toolboxes that were previously hidden.
So it might not be one single viral ancestor for all viruses, but perhaps a shared pool of very ancient protein building blocks that different viral lineages have drawn upon over time.
That's a very insightful way to put it. The ancient cellular origin model we discussed earlier provides a framework for reconciling these seemingly different perspectives. Viruses might not be monophyletic in the sense of all coming from one specific viral ancestor, but their diverse lineages could have emerged from this common pool of ancient cellular precursors. This is different from the idea of numerous completely independent origins from modern cellular components, as a strict escape hypothesis would imply for all viruses. So it's a story of deep, shared ancestry at a fundamental molecular level, even if there wasn't a single first virus that gave rise to all others.
It's like they all have distant cousins in this lost world of ancient cells, even if they don't have one immediate common viral parent. Now, the source touches on an interesting tension looking at viruses as integrated processes within larger biological units called holobionts versus trying to define their distinct evolutionary origins. Can you elaborate on that a bit?
This is a really crucial point in our understanding of biology, yeah. The holobiont concept emphasizes that an organism and all the microbes living in or on it, including viruses, can function as a single integrated biological unit. Viruses, through their interactions and their ability to transfer genetic material, are clearly deeply woven into these systems. However, this functional integration at the ecological level doesn't negate the importance of understanding their evolutionary history. We still need to trace their origins, even if those origins are complex and interconnected due to things like horizontal gene transfer. So, while viruses are undoubtedly integral to the biology of their hosts in many ways, understanding where they came from remains a fundamental question in evolutionary biology. It's about understanding both their deep historical roots and their current ecological roles within these larger biological systems.
Okay, so viruses have a complex and possibly very ancient history. Now let's shift our focus to the other major player in our story, the rise of eukaryotic cells. Our source describes this as a major leap in complexity happening quite a long time ago, somewhere between, 1.8 and 2.7 billion years. Why is this timeline so significant?
The timing is absolutely crucial because it sets the stage for when these interactions could have started to significantly influence the evolution of eukaryotic cells. The advent of eukaryogenesis with the emergence of the nucleus, mitochondria, the endomembrane system, the cytoskeleton, it created a completely new and far more complex cellular environment for viruses to interact with. Any major viral contributions to these fundamental eukaryotic features would have had to occur within this time frame. So the timeline tells us when the opportunity for these profound interactions arose.
And the source highlights the cornerstone event of mitochondrial endosymbiosis, when an archaeal host engulfed an alpha proteobacterium that eventually became the mitochondria, our cellular power plant. That's a pretty well-established part of the story. But what about LSCA, the last eukaryotic common ancestor? That single-celled organism that's the ancestor of all modern eukaryotes. It was relatively simple.
Not at all, apparently. The current scientific understanding is that Lesse was already surprisingly sophisticated. It had already acquired mitochondria, a nucleus, a complex endomembrane system, and a dynamic cytoskeleton. And what's particularly relevant to our discussion today is that Lesse's entire protein repertoire, its proteome, appears to have been a mosaic, a chimera. Genetic contributions not just from its archaeal host and the bacterial endosymbiont, but also from other bacteria through horizontal gene transfer. And remarkably, as our source highlights, a very recent analysis from October 2024 suggests there were also multiple waves of HGT from a diverse range of viral donors, some of which might have even occurred before the mitochondrial endosymbiosis event. So even at the very base of the eukaryotic family tree, viruses seem to have already been adding to the genetic mix.
Wow, so even at the dawn of eukaryotic life, viruses were already contributing. That really changes the perspective. From just seeing them as threats to potential early collaborators, or at least significant genetic influencers. And the source emphasizes that the key innovations defining eukaryotes likely happened during stem eukaryotic evolution, that period after the divergence from Asgard archaea, but before LECA. Why is this period so important?
That stem period is critical because it's when the defining characteristics of eukaryotic cells were likely taking shape. It's also a somewhat enigmatic chapter in evolutionary history, often represented by a long branch in phylogenetic trees, meaning we don't have many clear fossil intermediates to study directly. It makes reconstructing the exact sequence of events challenging. However, it's within this relatively obscure evolutionary window that the proposed viral influences on core systems, like the nucleus or DNA replication machinery, would have had to occur if they were indeed fundamental. So if viruses played a role in these key innovations, the STEM period of proto-eukaryotic evolution is where we'd expect to see their fingerprints.
So eukaryogenesis wasn't this neat linear progression, but more of a dynamic, maybe even chaotic mixing and matching of genetic material from various sources, including, significantly, viruses. That makes a lot of sense given how abundant viruses are and their known ability to transfer genes.
Exactly. The emerging picture is of a dynamic series of symbiotic integrations and horizontal gene transfers, a real evolutionary melting pot. Given their sheer abundance and their established role as major agents of HGT, viruses would have been prime candidates for contributing genetic material and novel functions during this pivotal period. The chimeric nature of LEGA's genome really underscores the high probability of significant viral contributions right from the start.
Okay, so viruses were present and likely interacting with early eukaryotes. Now let's delve into some specific ways our source suggests viruses might have actively shaped eukaryotic evolution beyond just being parasites. The first one is truly fascinating. The transition from an RNA world to the DNA-based genomes we see today.
This is a really thought-provoking hypothesis, yeah, largely championed by Patrick Forterre. The core idea is that DNA itself, along with the complex enzymatic machinery needed to replicate it, might have actually evolved first in viruses. One potential reason. DNA would have offered a more stable and less vulnerable form of genetic material against host enzymes that degrade RNA. Or maybe it was a way for viruses to distinguish their own genetic material from the host's RNA. So in this view, DNA was initially a viral innovation.
So DNA, the very blueprint of our genes, could have been a viral invention, like an evolutionary defense mechanism that then spread. And how did it get into cellular life?
Forterre proposes that these novel DNA-based replication systems could have been transferred from viruses to cellular hosts on multiple independent occasions. This could potentially explain why we see significant differences in the DNA replication machinery of bacteria compared to archaea and eukarya, which share more similarities in this area. The idea is that different viral lineages might have independently introduced DNA and its associated tools into different cellular lineages. It's like different tradespeople bringing different sets of advanced tools to the construction site of life.
That's a mind-blowing concept, that the fundamental molecule of our heredity might have viral roots. And our source mentions that many DNA viruses today encode their own DNA replication enzymes that show closer similarities to the eukaryotic and archaea versions than to the bacterial ones.
Precisely. For example, the replicative DNA polymerases in eukaryotic cells, like DNA polymerase delta, show more structural and sequence similarities to those found in certain large DNA viruses, like phycodinaviruses and the T4 phage, than they do to the primary replicative polymerases of bacteria. This phylogenetic kinship lends support to the idea of a viral contribution to the eukaryotic DNA replication toolkit.
But, as our source rightly points out, this idea isn't universally accepted. There are alternative theories, right, about how DNA might have originated within cells themselves.
That's correct. Some researchers argue that the complexity of establishing an entire DNA replication system from scratch in a virus without a more extensive cellular context presents a significant challenge. Others point to plausible chemical pathways for the gradual evolution of DNA from RNA precursors within a cellular environment. The debate is ongoing. Further research into DNA replication mechanisms across all domains of life is essential to fully unravel this ancient puzzle. So, while the idea of viral origins of DNA is provocative, it's still an active area of research and discussion.
Absolutely. And even with DNA taking center stage, RNA is still critical, so it's clearly complex. Now let's move on to perhaps the most radical idea discussed, the viral eukaryogenesis, or VE, hypothesis. This proposes a virus played a central role in the origin of the eukaryotic nucleus itself. That sounds like a real curveball.
It is a truly radical hypothesis, yes, championed by researchers like Philip Bell, and it certainly generates a lot of discussion and debate. The core idea is that the eukaryotic nucleus didn't gradually evolve within an archaeal cell, but instead originated from a complex viral replication factory. Imagine a large DNA virus, maybe an ancestor of the nucleocytoviricota, the group, including giant viruses and pox viruses, establishing a long-term persistent infection within an archaeal host cell.
So the virus basically sets up its own dedicated command center inside the archaeal cell.
Exactly. The virus would have reprogrammed the host machinery to support its own replication, creating a dedicated, membrane-bound or associated compartment to house its DNA and carry out replication and transcription. Over vast evolutionary timescales, the argument goes, this viral factory gradually evolved into the eukaryotic nucleus, and the viral genome eventually became the primary genetic material within that nucleus. In this view, the eukaryotic cell is a sort of superorganism composed of at least three key players: the archaeal host, the bacterial ancestor of the mitochondrion, and the nucleus-forming virus. It's a truly symbiotic origin story on a grand scale.
That's a complete reimagining. What kind of evidence supports such a bold claim?
Well, there are several striking parallels between modern large DNA viruses and the eukaryotic nucleus. These viruses create intricate viral factories that, much like the nucleus, are often enclosed by or associated with membranes, serve as the primary site for DNA replication and transcription, and even separate transcription from translation. Many large DNA viruses also have linear chromosomes, like eukaryotes, and possess mechanisms for capping their messenger RNA, also characteristic of eukaryotes. Furthermore, as we touched on with giant viruses, some of them encode genes for functions we typically think of as purely cellular DNA replication, repair, transcription, even bits of translation machinery. The example of Medusa virus is particularly intriguing. It contains genes for histones, DNA polymerase, and RAN GTPase, a key protein for nuclear transport in eukaryotes, and shows evidence of gene transfer with its host.
So these giant viruses seem to possess some key nuclear characteristics, and the VE hypothesis extends beyond just the nucleus itself to other eukaryotic features.
Yes, it does. Proponents suggest mitosis, the process of chromosome segregation, might have evolved from viral mechanisms for distributing replicated viral genomes. Eukaryotic nucleosomes, the DNA packaging structures, are proposed to have originated from viral DNA packaging proteins, and indeed, some giant viruses have histone-like proteins. Some researchers even speculate that meiosis and sexual reproduction might have roots in viral processes involving genetic exchange, maybe initially viral and mitochondrial genomes.
What about the cytoskeleton, the cell's internal scaffolding, and the endomembrane system, that network of internal membranes? Do they fit in?
The VE hypothesis proposes the ancestral virus might have introduced a tubulin-based cytoskeleton initially used for organizing the viral factory. Similarly, viral membrane bending proteins used for forming the virus particle or factory could have been precursors to the eukaryotic endomembrane system. The observation that some bacteriophages can create nucleus-like structures within bacteria, complete with spindle-like structures, shows that viruses do have the capacity to organize intracellular space in sophisticated ways.
It's a really compelling, almost sci-fi narrative. But our source also points out it remains highly controversial. What are the main sticking points?
The biggest challenge is definitively proving direct evolutionary relationships, clear homology, and tracing a precise pathway from a hypothetical ancient viral factory to the incredibly complex eukaryotic nucleus, with things like nuclear pore complexes. The exact nature of the hypothetical ancestral virus and the specific archaeal host involved also remains largely speculative. It's a fascinating idea that pushes boundaries, but more direct, concrete evidence is needed for broader scientific consensus. However, the ongoing study of modern giant viruses definitely lends plausibility to the idea that ancient viruses could have been involved in such transformative interactions.
Absolutely. The sheer complexity and gene content of giant viruses make you consider the possibilities.
Oh,
Now let's move on to a mechanism of viral influence that's more widely accepted: horizontal gene transfer, or HGT. We know viruses do this a lot in prokaryotes. How significant has it been in eukaryotes?
Oh, undoubtedly significant. Viruses have been major agents of HGT to and between eukaryotes throughout their history. They use various mechanisms: transduction, integrating their own genomes into the host genome as proviruses or EVEs, and capturing and retransmitting genes between hosts. That recent analysis of the LSEA proteome we mentioned, revealing multiple waves of viral HGT, some possibly pre-mitochondrial, really emphasizes the early and substantial role of viruses in shaping proto-eukaryotic genomes.
So not just the occasional stray gene, but potentially a significant sustained influx right at the base of the eukaryotic tree. What kind of impact could this have had?
Viral HGT can promote genetic modularity, allowing for the rapid exchange of functional gene units. This can lead to quick evolution of new viral capabilities, and if these modules get stably integrated into eukaryotic genomes, they can introduce entirely novel traits, new metabolic pathways, stress responses, DNA replication components, even host manipulation tools. It's like viruses were constantly providing new genetic Lego bricks for early eukaryotes to experiment with.
Our source gives a really compelling example. Telomerase reverse transcriptase, TERT, crucial for our chromosome stability. Where did that come from?
Phylogenetic studies strongly suggest that TERT, the catalytic core of telomerase, originated from a reverse transcriptase encoded by retroviruses or other mobile retroelements. These are genetic elements that use RNA to synthesize DNA, like retroviruses. Telomerase solves the end replication problem for linear chromosomes, and its evolutionary link to viral mobilome reverse transcriptases is a powerful illustration of how a fundamental eukaryotic function may have been co-opted from the viral world. So yes, a key process in our own cells likely has viral ancestry.
It's amazing. A key to our genome integrity has roots in these mobile elements, potentially including viruses. So it really appears viruses weren't just parasites, but active contributors to the eukaryotic toolkit.
Precisely. The very nature of viral parasitism, requiring such intimate interaction with host machinery, ironically created numerous opportunities for hosts to capture and repurpose viral genetic innovations, turning potential threats into evolutionary advantages. It's a fascinating example of conflict driving innovation.
Now, we've mentioned giant viruses several times. Our source has a dedicated section on them. What makes these unusual viruses so particularly important here?
Giant viruses, with their huge genomes and particles encoding genes once thought exclusively cellular, have really revolutionized virology. While the consensus is they likely evolved from smaller viral ancestors via gene acquisition from hosts and microbes, not direct cellular reduction, their existence shows the remarkable genetic complexity viruses can achieve. Their ancient origins, likely coexisting with early eukaryotes, and their broad range of protist hosts strongly suggest they've been interacting with and influencing eukaryotic evolution for a very long time.
And their impact isn't just their own infection cycle.
Absolutely not. Giant viruses are potent agents of large-scale HGT. They can shuttle substantial DNA into host genomes, potentially significantly shaping their evolution. Plus, they play crucial ecological roles regulating microbial eukaryote populations, likely doing so for billions of years, indirectly influencing host evolution that way too.
And then there's that truly fascinating interaction with virophages, viruses that infect giant viruses, like viruses all the way down.
This is a remarkably elegant example of intricate ecological and evolutionary relationships. Virophages are essentially hyperparasites of giant viruses. What's really interesting is that virophage genomes can sometimes integrate into their Protestant host genomes, becoming EVEs. If that host cell later gets infected by a specific giant virus, these integrated virophage sequences can be reactivated and interfere with the giant virus replication, protecting the host. It's a case of the host co-opting a viral element for defense against another virus. The genetic diversity in giant viruses themselves also points to this ongoing coevolutionary arms race.
It's like a nested system of viral interactions intricately shaping the host. Which brings us to the final point. This fundamental idea of perpetual coevolution, a constant arms race.
It is a relentless and ancient dance, often described by the Red Queen hypothesis, where both virus and host must continuously evolve just to keep up. This constant pressure has driven the development of sophisticated host defenses, like antiviral proteins and RNA interference, and equally sophisticated viral countermeasures. And as we've discussed, the integration of viral material as EVEs is another key outcome, with some EVEs being repurposed by the host, even as defenses against related viruses.
So it's not just conflict, there's integration, even a form of cooperation arising from this long interaction like the virophage defense. And this has been going on for billions of years.
Exactly. The evolutionary histories of major eukaryotic virus lineages show clear hallmarks of this long, intimate coevolution. The modular nature of viral evolution, driven by HGT and recombination, allowed them to rapidly adapt and diversify within the expanding eukaryotic domain. When we consider the origins of the eukaryotic cell itself, the symbiosis between archaea and bacteria, it's becoming increasingly apparent we should also consider viruses as potential, perhaps equally crucial, partners. Their contributions, though varied and often less obvious, may have been just as essential in shaping the very cells that constitute all complex life, including you.
This has truly been a deep dive. So to synthesize all this compelling information, what are the most significant key takeaways?
The accumulating evidence really points towards a fundamental shift. Viruses appear to have been profoundly influential, not just minor players. We see strong evidence of significant HGT contributing to LECA's makeup, possibly even pre-mitochondria. There are compelling, though still debated, hypotheses suggesting direct viral roles in major innovations like DNA origin, the nucleus, telomerase, and the ongoing coevolution has been a major driver of host defense evolution and a source of genetic novelty via EVEs.
So if viruses were key players in fundamental innovations, then viral evolution itself becomes a primary force shaping eukaryotic complexity,
Yeah.
not just cells evolving alone.
Precisely. A major viral contribution, like a nucleus precursor, could have had cascading effects on host cell evolution, creating new pressures and opening up entirely new possibilities.
And while there's growing consensus on the antiquity of viruses, their role in HGT and coevolution, there are still major unanswered questions.
Absolutely. The precise origins of diverse viral lineages are still debated. The VE hypothesis, while fascinating, remains controversial. The exact timing and nature of viral contributions to early life pre-ELUCA are largely speculative. We also face huge knowledge gaps. The vast viral dark matter, unknown functions of many viral orphans, reconstructing ancient HGT events, especially during early eukaryogenesis, and we still have much to learn about the proto-eukaryotic hosts themselves and their viral interactions during that critical stem period.
And the traditional tree of life. It might need a significant rethink given all this viral influence and gene exchange.
Yes. The increasing recognition of widespread HGT, especially via viruses, suggests the early tree of life might be more like a complex network or rhizome. Viruses are crucial connectors. The concept of a broad viral supergroup further complicates the traditional tree representation. How do viruses fit into the overall picture? It's a big question.
So what are some of the most promising avenues for future research to help unravel these ancient relationships?
Methodological advances are key. Continued use of structural phylogenomics, leveraging tools like AlphaFold, will help uncover deeper relationships. More extensive comparative genomics from metagenomic sequencing of diverse environments and undul-sampled hosts is crucial. Studying ancient EBEs provides direct fossil evidence. Specific focus areas should include viromes of Asgard archaea, functional characterization of viral or R-fans, experimental evolution studies, and further exploration of giant virus biology. Ultimately, it requires truly interdisciplinary approaches integrating molecular biology, genomics, ecology, and deep evolutionary theory. Virology is central to understanding life's history. We also need more sophisticated theoretical models that embrace the reticulate nature of ancient life, moving beyond simple tree thinking.
This has truly been eye-opening. The core message seems to be that viruses are far more than just agents of disease. They appear to have been deeply influential in eukaryotic evolution right from the start.
Absolutely. From contributing to the genetic toolkit of the first complex cells to potentially playing pivotal roles in originating fundamental features like DNA, the nucleus, and telomerase, viruses appear crucial. And their ongoing coevolution continues to be a powerful force shaping the biological diversity we see today.
So as we bring this deep dive to a close, I want to leave you, our listener, with this final thought. Consider how our understanding of life's history fundamentally shifts when we recognize viruses not merely as threats, but as fundamental creative forces that have actively shaped the very cells we are made of. What other long-held biological assumptions might need reevaluation in light of these discoveries? If this exploration into the intertwined destinies of viruses and eukaryotes has sparked your curiosity, we definitely encourage you to delve deeper into the source material or explore the rapidly evolving research landscape. There's a whole universe waiting beneath the surface.